Come Giovanna ha detto, sono attualmente un lecturer in geostatistics all'Università di Lancaster, in questo gruppo che si chiama CHICAS, dove lavoriamo un po' all'interfaccia tra epidemiologia, statistica e informatica. Quello che faccio io nel mio ambito di ricerca in generale è statistica spaziale e spazio-temporale applicata a malattie infettive, anche malattie non comunicabili. Mi interessa anche molto di server design per dati spaziali e spazio-temporali, creazione di sistemi di sorveglianza in tempo reale, soprattutto durante gli ultimi 2 anni con il Covid, e ultimamente ho iniziato a interessarmi sempre più su come siccome lavoriamo tanto con istituzioni come WHO e quindi i nostri risultati sono importanti dal punto di vista di policy making, anche cercare di creare delle user web interface che siano user friendly per permettere a end user di utilizzare questi approcci che sono complicati per una persona che non è il mestiere, non è uno statistico, però i cui risultati sono importanti per informare decisioni dal punto di vista dei trattamenti per queste malattie. Oggi parlerò di un modello che ho sviluppato per modellare in modo congiunto la prevalenza di una malattia che si chiama filariasi linfatica, quindi voi probabilmente ne avranno sentito parlare, che di solito viene misurata con due approcci diversi e quindi quello che succede è che abbiamo due fonti diverse di informazioni che sono correlate tra di loro e vogliamo sfruttarle nel modo migliore possibile. Ora, se non vi dispiace, passerò all'inglese. So I'll start now with a very small introduction about lymphatic filariasis. This is one of many vector-borne and neglected tropical diseases. I work a lot with neglected tropical diseases, and it's a class of diseases that are neglected because since they don't cause a lot of mortality, the main issue is morbidity. Then there is a lot of time there is not a lot of interest, like you have for malaria, for example, and there is also not a lot of funding for treating this disease and doing research in this field. However, over the last years, there have been more and more investment in this field, so we are starting to see a lot of developments in terms of diagnostics, in terms of tools that can be used to tackle these neglected tropical diseases. And lymphatic filariasis has been targeted by the WHO as one of the targets is to eliminate as a public health problem by 2020, although most countries didn't reach this goal. And the other target by 2030 is eliminate lymphatic filariasis in terms of transmission. Because WHO usually distinguish between elimination as a public health problem, that means taking the prevalence down to a level that doesn't cause a lot of morbidity, and elimination as transmission, that means that the prevalence is so low that the vector cannot transmit the disease anymore. And by 2030, the goal would be to eliminate this disease in most countries currently endemic for LF, eliminate transmission. The vector is mosquitoes, 3 species of mosquitoes, Aedes, Anopheles, or Culex, and you find one of them depending on what part of the world you live. And what this disease causes is a disruption in how the lymphatic vessel function, and this can lead to severe chronic conditions like adrocile lymphedema and elephantiasis. These 2 pictures below, the one on the left shows the parasite, the worm, that the mosquito will transmit to the human, while on the right is an example of elephantiasis, one of the debilitating conditions that you can develop if you're afflicted with lymphatic filariasis and is not treated as soon as possible. I want to tell you a little bit more about the transmission cycle, how a human gets infected with lymphatic filariasis. And everything starts with an infected mosquito that when bites the human will pass the larvae of the parasite that goes into the skin and moves into the vessel. When the parasite larvae is in the vessel, then it starts to develop, becomes an adult worm. And if you have both a male and female adult worm, they will start to reproduce, generate some offsprings that are called microfilariae. And these are the ones that cause the infection in the vessel. Okay, so that is why we usually don't talk about filariasis prevalence, but we talk about microfilariasis prevalence. And then if another mosquito bites the infected human, takes from the blood the microfilariae, that then goes to another larval stage within the mosquito, and then the transmission cycle goes on and on. In terms of the current spatial distribution of this is a disease that can be found in low and middle income countries that have a tropical climate, because this type of mosquitoes can only live under certain climatic conditions. It means tropical conditions, so only under this condition you can find the mosquito that cause this, transmit this disease. Africa is mostly afflicted and is essentially the area that we work mostly with, but you can also find some areas in India, close to India, and also in South America, there are still some countries that are endemic with lymphatic filariasis. Now, what to say a little bit about prevalence surveys and decision making, because we work, this work is based on prevalence surveys that collect data on LF, on lymphatic filariasis. And a lot of countries currently are supported by the WHO, the World Health Organization, that helps them managing control and monitoring programs for several neglected tropical diseases. And the very first stage of this program is to conduct something that is called a mapping survey. Now, the goal of a mapping survey is, before doing any treatment, trying to assess what is the current burden of the disease, and then based on that, we can decide how much treatment we should give to a certain area, or if it should be treated at all. The type of data that is collected is this triplet. So we usually have locations of the place that we have visited, that I'm calling X. These are fixed locations that we need to allocate based on some survey design approach. And then at each of those locations, this usually can be villages or school. For lymphatic filariasis, the surveys are at community level, so we go to the village. Then we test a subset of individuals and within those locations, and we record the number Y of cases of individuals that were tested positive for lymphatic filariasis. And then we need to use some model to infer what is the true underlying level of prevalence and assess the endemicity status, and based on this, decide what is the appropriate level of treatment. And understanding also how the prevalence varies over space is very crucial to inform disease control policy for LF, because remember that we work in a setting where we have very limited resources. So we want to optimize them as much as possible, and we want to do treatment that is spatially targeted. So we don't want to overtreat or undertreat some places. We want to provide the correct level of treatment for several areas of interest. Now, when we conduct this survey for LF, how do we test people? And thinking about the diagnostic tool that is being used is crucial when we build a model. There are currently two approaches. One that is the parasitological approach, and this essentially consists in taking a blood sample from the individual and then counting the microfilariae, the worms, the offsprings, under the microscope. The tricky thing about this is that it's very time consuming and it's expensive. And moreover, since one characteristic of the microfilariae is that they have a sort of night life cycle, so to find them, you need to take the blood sample during night. So that is another complication. Starting from the end of the 90s, beginning of 2000, they developed a rapid antigenetic test, this immunochromatographic CAR test, ICT, that essentially is a rapid antigen test that measures the presence of adult worm antigen that circulates in the blood. And what happens is that from 2000 on, most prevalence surveys will be conducted using this new approach. So there are serological surveys rather than parasitological survey. The problem is that the target of interest, the measure of interest, is MF prevalence rather than antigen prevalence, because the WHO threshold for treating and elimination, they're all set up and parameterized according to MF prevalence. And also there are a lot of MF surveys out there, so you don't want to throw them out. So the issue is now, how do we combine these two sets of data together in a consistent way to try to get the best out of this? So type of data that we're going to work with is prevalence data that have been collected with two different approaches. We're going to have prevalence data for antigen, so when we use ICT testing, and prevalence data for MF. And the situation will usually be that the first set of data, so the serological survey, are much more common. So you have a much bigger sample size for ICT data rather than for MF data. Now, an obvious model for prevalence data is using a logistic model. So we assume that the number of individuals tested, either with one of the two diagnostic tests, comes from a binomial distribution with the denominator equal to the number of people that we test. And then this P indicates at a certain location X what is the probability of being tested positive with either of the two tests. So we can start with a very simple logistic model, but now the question is, first, how do we exploit the correlation between the antigen prevalence and the microfilariae prevalence? Because although they won't be identical, they will be correlated. From a purely biological point of view, they have to be correlated. So we want to exploit that relationship and understand what that relationship is. And second, since the vector, the mosquitoes, is not just uniformly distributed over space, the abundance of the vector depends on a lot of environmental characteristics. So the spatial distribution of the vector, so the distribution of the vector is spatial as a spatial component. And that will induce also spatial variability in the prevalence. So we also want to exploit the spatial correlation present in the data. So essentially, if I observe at a certain location a very high prevalence of ICT, if I go to a village close by and I measure MF prevalence, I would expect also MF prevalence to be quite high, because prevalence is correlated and the prevalence measure with the two diagnostic is correlated as well. So the main goal will be to build a model that is able to exploit the abundance of data that we have for ICT prevalence surveys, the relationship and the relationship that, biological relationship that there is between ICT and MF prevalence, with the final goal of predicting microfilariae prevalence at unobserved locations. Because remember, we have limited resources, so we cannot do a census of the population. We can only sample a finite amount of villages and test a finite amount of people. And then based on that information, we want to generate a predictive maps, and we want to be able to predict anywhere in any area of interest. So a little bit now about methodology. The approach that I'm going to use, I'm calling this a semi-mechanistic approach because I'm going to exploit the biological mechanics behind the way a person is infected and how the worms reproduce in the body to build this model. But then I will also add some more empirical components to this approach. So that's why I'm calling this a semi-mechanistic approach. So I'm going to start to define some quantities. I'm going to define with W the number of adult worms in a sample individual, and with M the number of offsprings, so the MF count. Now, one assumption that we can do is we can say that the number of adult worm in sample individual is Poisson with a certain rate, lambda, that we don't know and we want to infer. And then conditional on the number of adult worm that you have in your body, you will have also, you can expect to have a certain number of offsprings based on this parameter that I'm calling alpha, that is the reproductive rate of the adult worms. So we have two Poisson distribution. The second one, so the number of offspring, is conditional on how many parents, how many adult worms we have in the body. From this one, now I want to work out what's the probability that I'm going to be tested positive And for ICT, if I use ICT, remember I'm counting the antigen, and you're going to have antigen if you have adult worms in your body. So if you have a positive number, a number of adult worms greater than 0 in your body, then you're going to be tested positive with ICT, and also take into account for the sensitivity of the ICT test because it doesn't have a perfect sensitivity. So what I want to work out based on these two distributions now is the probability that I will find a positive number of adult worms in a sample individual. Since this is Poisson, then this has a closed form and is very easy to compute. We can do the same for MF. So also in this case, it has positive, you have a positive number of microfilariae in your body. You need to marginalize this distribution, so integrate out W and compute the marginal of M. That, likely for us, is again another Poisson, and you get this other closed form expression. So what I'm going to do now is going back to my binomial model, and when I define these two probabilities, this is the functional form that I'm going to use for these two probabilities. And also, both of them, they depend on lambda and also alpha, link the two probabilities of testing positive and the two prevalences. And this gives me a way of linking up those two prevalences that is based on the underlying biology. There is, I also said before that I want to also take into account the fact that the intensity of the worms and the chance of finding worms varies over space because we have different transmission intensity based on the abundance of the vector. And a limitation to this approach is that what I'm assuming at the moment is that the worm intensity lambda is currently homogeneous over space. Okay, it doesn't vary over space. But if we want also to write a spatial structure, what we can do is then, instead of having lambda that is a constant parameter, now I build another kind of sub-regression model for that lambda, and I make lambda a stochastic process, in particular a spatial stochastic process. Now, model lambda as a function of x, the location, so now it's a function of space and varies over space. And I model this on the log scale because lambda has to be positive. Okay, you cannot have a negative number of worms. So by applying the log transformation, I know that I can work now on the real line, and then I can transform back to positive numbers. And I'm going to model now the worm intensity as a function of three components. So first of all, I'm trying to explain the spatial variability in that worm intensity using a set of covariates that I'm calling D that are also spatially defined. So this will be a set of environmental variables, might be temperature, rainfall, other environmental conditions, bioclimatic factors, and so on, that we know have a relationship with the distribution of the vector. The second term is S. This is a spatial Gaussian process. So what that means is a set of random effects that are spatially structured, and it is used essentially to absorb any residual spatial variability that I cannot model with the covariate that I've included in my model. So sometimes I like to think about this as a proxy for our ignorance, okay? We don't know what is causing this spatial structure in what we see, but we can put something there, a term, that is able to absorb that residual spatial variation that we cannot explain with our covariates, with our predictors. And from a mathematical point of view, this term is just a set of multivariate normal with a variance-covariance matrix that is parameterized by distance between locations, and I'll talk now a little bit more about how we parameterize that function of distance. The last term, Z, this is another set of random effects, but these don't have any spatial structure, they're just independent. So in the same way as S is capturing any residual spatial variation, Z will capture any residual variation that doesn't have a spatial structure or any sort of structure. And these are defined as a set of independent and identical distributed normal random variables with mean 0 and variance tau square. So now I've defined my model, and oh yes, the last thing I wanted to do is that these two equations that we have defined before, that allow me to build a relationship between the two prevalence, the prevalence of ICT and the prevalence of MF. And what is known in biology is that when you measure the antigen, you tend to always overestimate the actual prevalence, actual MF prevalence. And the reproductive rate alpha controls what is the actual shape of that relationship. So also the formula that we obtain by making that sort of reasoning before is in line with what we expect to see in reality.
So ICT overestimates it.
Yes, always, almost always overestimates on average MF prevalence. Yes. Now, when we define this spatial Gaussian process, we need to define a correlation function that is a function of distance and tell us essentially how strongly correlated are two observations that are far apart at a certain distance. Now, this type of correlation function usually have the characteristic that the closer you are, the higher will be the correlation, and the farther you will be in space, the less will be the correlation, of course, because things that are measured very distant from each other, they don't tend to be similar. And there is this general class of correlation function that have certain property that we need to fit these models that are called modern correlation functions, and are managed by two parameters. One is this kappa parameter that determines the smoothness of the underlying process. Because this spatial Gaussian process, essentially what it is, allows us to fit a kind of continuous smooth surface for the prevalence. And this parameter manages how smooth that surface will be. The other important parameter is called phi, and it's called the range parameter. This will tell you how strong is the correlation in your data, the spatial correlation in your data. These two parameters cannot be estimated simultaneously because they're not both identifiable. And what is done in practice is to fix kappa, also because this kappa won't make a huge difference in your results, to be honest. So you fix kappa or you try different values for kappa. There are two or three common values that identify three main type of correlation functions. But then what you essentially want to estimate is this parameter phi that controls the strength of the spatial correlation. And a good way to interpret this parameter then is to work out what is the practical range, that is the distance at which the correlation is very close to zero. So if your scale parameter is, for example, 10 kilometer, and you're using exponential correlation function that you get for kappa equal to 0.5, then that will mean that your data are correlated up to a distance of approximately 30 kilometers. So that is the type of interpretation that we do for that parameter. Now, this is the most complex part, and is how to make inference about this model. I'm going over this very quickly, and please ask me for more details if you want later. But the parameters now that we need to estimate is this vector of parameter theta that is comprised from the reproductive rate alpha. These are the regression coefficients for the environmental variables that we include in the model, the variance of our spatial Gaussian process, the range parameter that controls the strength of the correlation, and then the variance of the non-spatial random effects. And our assumption is that conditionally on lambda, that is our latent process, my observations are a set of independent binomially distributed random variables. To write down the likelihood of that model is a little bit complex because we cannot have it in closed form. And what we need to do is to, the likelihood will be essentially the joint distribution of this y. Calculate that joint distribution. Yeah, there was a question? Okay. So to calculate that joint distribution, what we do is to first express the joint distribution as the distribution of the data conditional on the process times the marginal distribution of the process. And if we then integrate out the latent process, we get the likelihood. This doesn't have a closed form, cannot be calculated, so you need to do some mathematical trick to arrive to an expression that is that we can calculate somehow. This is just some rearrangement, but the point here is that we can approximate this likelihood function as a ratio between these two quantities, that is the marginal distribution of the latent process according to the set of parameters that we want to estimate, and then this theta zero, that is an initial guess of what we think is the true parameter for these coefficients, times the conditional distribution of the latent process given the data. Now, if you see something like this, you can use a Monte Carlo approximation to calculate this integral only if you're able to sample values from lambda. So you can sample, if you're able to sample from this conditional distribution, then can you see my mouse? Yes, okay. If you're able to sample from this conditional distribution, you take a lot of samples, and then you can empirically compute this ratio and calculate the maximum likelihood estimate for your parameter theta. And that is exactly what we do. So we use Markov chain Monte Carlo to sample from this distribution, because also this doesn't have a closed form. We simulate a lot of samples that I'm going to call lambda b from this distribution, and then we approximate the likelihood with its Monte Carlo counterpart. Okay. So we take the samples and we compute the average ratio, and that, as b goes to plus infinity, then will give you exactly the likelihood that we want to estimate. And of course, this is an approximation and relies on some assumption. You need to be sure that you're using a good MCMC algorithm to sample from this conditional distribution. And the other important thing is that the quality of this approximation depends on what is how good is your initial guess of the parameters, because remember, you need to define this theta zero to use this this approach that is called Monte Carlo maximum likelihood approach. Now, a strategy that you can use is to repeat this estimation several times per essere sicuri che convergiamo ai veri valori di maximum likelihood. Quello che si può fare è ripetere questo processo diverse volte. Quindi io prima definisco delle guess, dei parametri iniziali per θ 0, e con questi parametri iniziali fit il modello e ottengo una prima stima di maximum likelihood. Dopo uso questa stima di maximum likelihood che ho ottenuto dalla prima iterazione come il mio prossimo θ 0. Quindi di nuovo ripeto il mio algoritmo e vado avanti fino a che vedo che non si stabilizza e questi parametri non cambiano più. Quindi questo è un modo un po' per garantire che anche se partiamo, diciamo, un po' da lontano e la nostra guess iniziale non è ottima, comunque arriviamo a delle stime che sono reliable. Ok, quindi basta con la teoria e le equazioni. Abbiamo costruito il nostro modello, sappiamo come fare l'inferenza del nostro modello, ora vi mostro un'applicazione. In questo caso sto utilizzando dati che vengono dal West Africa, da 4 paesi: Benin, Burkina Faso, Ghana e Togo, e abbiamo quasi 1000 mapping surveys, quindi queste sono survey che sono state effettuate prima di fare qualsiasi trattamento. Okay? Quindi dovrebbero darti un'idea dello stato endemico della malattia. E poi abbiamo 300 survey che sono invece riguardano il microfilariae e sono survey serologiche. In verde su questa mappa ci sono tutte le MEF survey, mentre in blu sono le ICD surveys, e abbiamo un coverage spaziale che è decisamente migliore con le survey serologiche, ma non vogliamo buttare via comunque i dati che vengono da MF e vogliamo usarli per costruire quella relazione tra i due. Sulla destra invece questa qua è semplicemente la prevalenza che è stata misurata per queste survey. Qual è il nostro, diciamo, target per ottenere delle previsioni e il tipo di output che noi possiamo ottenere dal modello geostatistico che ho formulato prima? il tipo di output che otteniamo è la probabilità congiunta della distribuzione della prevalenza di MF a qualsiasi location che vogliamo, ok? Nell'area di interesse, condizionatamente a tutti i dati che abbiamo osservato. Ora, la cosa, diciamo, utile di non avere una stima di punto per le previsioni, un solo valore, ma di avere tutta la distribuzione di probabilità, tutta la predictive probability distribution, è che poi da quella ci possiamo calcolare qualsiasi proprietà, qualsiasi summary di quella distribuzione. Quindi se vogliamo poi calcolarci la media, la mediana, la varianza, o andare a vedere ad altri summary che sono più di interesse scientifico, possiamo farlo. Come ad esempio tante volte in questi casi quello che si vuole andare a vedere sono quelle che chiamiamo le exceedance probabilities, che sarebbe la probabilità che osservi, cioè che la prevalenza vera è più alta o più bassa di un certo threshold di interesse. Ecco, il fatto che la WHO ha dei threshold al di sopra o al di sotto del quale decidi se fare un certo tipo di trattamento oppure no, è importante cercare di dare i risultati in quei termini. E per noi è una proprietà di interesse andare a vedere quello che facciamo noi, perché questi, anche se creiamo delle mappe che sono continue, quindi facciamo previsioni a livello continuo, dividendo lo spazio in tanti piccoli pixel, le decisioni che vengono prese dalla WHO e all'interno di questi paesi sono a livello di queste aree amministrative che vengono chiamate implementation unit. Okay? Quindi anche se i risultati quindi grazie mille ancora per l'invito e per avermi ascoltato nell'ultima, non so quanto ho parlato, spero non troppo, mezz'ora.
Sì, mezz'ora.
Ok, bene. Grazie.